Hello everyone, I'm Andong Wang. This is our project, Interpretable Singular Value Decomposition Clutter Filtering in High Frame Rate Ultrasound Imaging. As we all know, SVD clutter filtering can effectively separate signals of tissue, blood flow, and artifacts for high frame rate ultrasound imaging. And there are various empirical approaches to estimate the separation by singular value selection. Specifically, given 3D ultrasound data D, we first reshape it and conduct SVD. After SVD, there are k singular values, and the estimation is to decide which group of singular values belong to tissue, blood flow, and artifacts respectively. However, currently, such estimation is done by trials and errors in an uninterpretable way. We still do not know which regions in an ultrasound frame each singular value accounts for. Therefore, our aim is to interpret clutter filtering and provide feedback to researchers to confirm singular value selection accuracy. First, let me explain why it is hard to interpret. When we call it clutter filtering, we hope to remove artifacts while identifying tissue and blood flow signals which locate in different regions. However, SVD separates data in the frequency domain. Look at the SVD formula. The first component has the highest spatial-temporal coherence, while the last component has the lowest. The GIFs below show that the first few components are more stationary than those of higher orders. Therefore, our method is to bridge the gap between the frequency domain and the spatial domain. We build the bridge by identifying the responsible region of each singular value. So, the next question should be, how we identify the responsible region of the I singular value sigma I. We get the responsible region by optimizing a 2D mask MI with 0 and 1 representing irrelevant and responsible signal samples respectively. Look at the figure. The yellow region represents the responsible region. It means that even when we eliminate other regions, the responsible region still preserves the key information of sigma I. To solve MI, we formulate an optimization problem. We first reshape MI to be a vector. Simultaneously, we get SI stars, which preserve all the singular values after sigma I. Next, we conduct element-wise product along the spatial dimension between MI and SI star, and conduct another SVD to the product result. Finally, the loss function. The loss function has three terms. The first is error term to minimize the error of responsible region. The second is derivative term to constrain spatial continuity. The third one is responsible region term to find the responsible region. It is based on assumption that when MI star correctly captures responsible region, so sigma 1 star U1 star V1 star transpose should approximate sigma I UI VI transpose. And the whole process is differentiable. We can backpropagate the loss and use SDG or LDM to solve the optimization problem. We evaluate our method on simulated ultrasound images and real high frame rate echocardiography data. The figures above show that our method accurately identified the spatial temporal coherence regions, respectively associated with components of different orders. Results on human echocardiography data show that the first few components focus more on background and clutter, and that higher orders pinpoint dynamic tissue regions. This agrees with our understanding of SVD filtering. To conclude, we propose a method to interpret SVD clutter filtering by identifying the responsible region each singular value accounts for. Such interpretation provides feedback to researchers to confirm component selection accuracy. However, the limitation of our method are hyperparameter settings may vary from case to case. Also, our method may not be able to interpret some singular values which are a mixture of artifacts and target signals. Thanks a lot.